Hello, class. So today we are going to talk about enzymes. And our key concept is that life depends on chemical reactions. Our essential question is, how do enzymes help metabolic functions? How do enzymes help metabolic functions? And as we see in this picture, we see a Venus flytrap, and it is eating a frog, or it is digesting a frog. So we want to talk about how our bodies process chemical reactions. So bonds break and form during chemical reactions. So our systems are constantly breaking and forming chemical bonds. So chemical reactions change substances into different ones by breaking and forming chemical bonds. Reactants are changed during a chemical reaction. So if I look here on these reactions, I have A plus B, and then I get this molecule AB. So in this instance, the bond is being made. I have AB, which gets broken down to A plus B. I can see here that this molecule is being broken down. I have AB plus C, which is going to yield AC plus B. So I see some chemical reactions displacing and reversing. And same thing for AB plus CD gives us AD and CB. So our products are made by this chemical reaction. So we have our reactants and then we have our products. Bond energy is the amount of energy that breaks a bond. So bond energy is the amount of energy that breaks a bond. Energy is added to break bonds. So let's take a look at this graph here. Notice that I have two graphs. I have one for an exothermic reaction and one for an endothermic reaction. Now, how we read this graph is this here on our x-axis is going to be time it takes. The y-axis is going to be the amount of energy utilized, or the potential energy. So energy is needed or is added to break bonds, which is known as our activation energy. So for example, if I start off with my reactants, I need this amount of energy to break up the reactants to make our products. Same thing here. If this is my reactants, this is the amount of energy needed. And at the end, that would be our products. Okay. Now, energy is released when bonds are formed. So we have energy is added to when bonds break, and energy is released when the bonds are formed. So chemical reactions release or they absorb energy. So you are either breaking something apart or you're putting something together. So either breaking something apart or you're putting something together. Activation energy is the amount of energy that needs to be absorbed to start a chemical reaction. So please make sure that you know this term, activation energy. Activation energy is the amount of energy needed that's going to be absorbed to start our chemical reaction. Okay, so this here would be our activation energy. And as we see, this man is pushing a ball up the hill. So the hill up here will be our activation energy. Obviously, once the ball gets released, then the energy gets released here as well. Right, in an exothermic reaction, we release more energy than they absorb. So exothermic reactions release more energy than they absorb. So like I said, please make sure you understand this term exo. Exo means like exit, right? Therm means like heat. Okay, so therm has to deal with heat. Sorry, my spelling is terrible up there. Okay, now reactants have higher bond energies than products. So the reactants have the higher bond energy than our products. What does that mean? So if I am going to, I have my reactants here. So I ate something. Now, because I ate it, I know that it needs to be broken down into the product. So let's take, for example, our lactose. Let's say you drank milk, right? And what happens is lactose needs to be broken down into these two products, which is going to be glucose and galactose. Now, in order for that to be broken apart, I need some energy to break apart the molecules into the products. So this is the amount of energy that's added. And then in the process, energy gets released. Energy gets released every time I break a bond. The excess energy is released by the reaction. So the excess energy is released by the reaction. Whereas in an endothermic reaction, we absorb more energy than they get released. So this is when you're making molecules for our system. So in this instance, endothermic reactions absorb more energy than they release. The reactants have a lower bond energy than the products, and the energy is absorbed by the reaction to make the difference. So how does this graph look differently compared to the graph before? Notice here that here's my line of our starting point. So here's my reactants. Notice now that the products being formed are above. So they're here above. It didn't go below as we've seen in an exothermic reaction. The exothermic reaction went down here, and then our energy got released. In this here, we absorb the energy. So the energy gets absorbed by the reaction to make the difference. All chemical reactions involve energy. So all chemical reactions that occur in our systems involve energy. Energy is either needed, it's absorbed, or it's given off and released during a chemical reaction. So we are either absorbing energy or we're giving it off. So we are either digesting or we are synthesizing, digesting or synthesizing. A catalyst lowers the activation energy. So catalysts are substances that speed up chemical reactions. And what they help to do is they decrease the activation energy and they increase the reaction rate. So going back to our example with milk. So if you are lactose intolerant, you don't have the enzyme lactase to break up milk. In that instance, your body would have a really hard time and would utilize a whole lot of energy, as we would see here, to break up lactose. Now, for those of us that have the lactase enzyme, notice here that when I add an enzyme, the energy here now from this point, as we see, has decreased. So I decrease the energy. What does that mean? So if I decrease the energy, that means that it doesn't take me a long time to break up the molecule. I then can form the products at a much quicker rate. So in this video, I'd like you guys to see it. It talks about how to speed up chemical reactions. Does a really good job of showing what are some factors that help speed up chemical reactions in our systems. All right, enzymes are biological catalysts. You need to know that. So each enzyme is a specific helper to a specific reaction. Each enzyme needs to be the right shape for the job, and enzymes are named for the reaction they help and usually end in the term ASE. So make sure that you know that. Enzymes end in ASE. So for example, lactose, which is milk sugar, gets broken down by lactase. Lactase is our enzyme for milk, or lactose. So for example, sucrase breaks down sucrose. Proteases break down proteins. Lipases break down lipids. DNA polymerase builds DNA. Now notice that not all enzymes break things apart. Some of them build. So I have these three enzymes break things apart. So if they are breaking things apart, these are going to be part of our exothermic reactions, because every time something gets broken down, energy gets released. Whereas this enzyme here, DNA polymerase, it builds DNA. So that would be an endothermic reaction. Endothermic meaning that since I'm putting molecules together, I am now going to hold the chemical reactions. I save them. Our enzymes are not used up. So anytime an enzyme is used, it does not fall apart or doesn't stop working. Okay. They are not used up. So once an enzyme breaks apart a molecule, it's then readily able to go break apart another one. So enzymes are not changed by the reaction. They're used only temporarily, and they are reused again for the same reaction with other molecules. There's very little enzymes that's needed to help in many reactions. So let's take, for example, like I said, here's lactase, and here's our substrate, which is called lactose. Now, the enzyme lactase is going to fit perfectly with the lactose sugar. Okay, it's going to bind at this molecule called the active site, So this here is the active site. The active site is where the substrate is going to attach to. Notice now that the enzyme forms pressures on the bonds, and then the bonds get broken, and now I made my two products. Once my products are released, my enzyme can then look for another lactose molecule and can break it apart. And they consistently are being used over and over and over and over again. They get recycled. Enzymes are known as a lock and key model. What that means is that the shape of the protein allows the enzyme and the substrate to fit perfectly, just like a lock and a key. There's a specific enzyme for each specific reaction. Okay. We make hundreds of thousands of different types of enzymes. And like we said, every enzyme that we make is going to be specific to the molecule that it breaks down or that it puts together. So notice here's sucrose. So let's say you had table sugar. You should have the enzyme sucrase. Sucrase is going to bind to the enzyme, sucrose in this instance. Water is added, the bond gets broken, and the substrate is converted to the products. So once I break sucrose down, I now form glucose and fructose, which are single sugars. Once these molecules are formed, notice now that my enzyme is available for another molecule or substrate, and it'll keep breaking up molecules until I have no more to break apart. All right, the enzyme vocabulary. So these are the terms that you need to know. An enzyme is a helper protein molecule. A substrate is the molecule that the enzyme works on. The products are what the enzymes help to produce from the reaction. The active site is a part of the enzyme that the substrate molecule fits into. So here's our active site, here's the substrate, here's the enzyme. All right, the shapes matter in terms of enzymes. Enzymes are proteins. Proteins do their job because of their shape. Anytime a protein unfolds or an enzyme unfolds, it destroys the shape. If it's the wrong shape, it can't do its job. The term for unfolding the protein is known as denature. It's known as denature. So there are two items that can cause an enzyme to not function. So one of them is going to be temperature, and the other one is pH. So temperature and pH play a big role as to whether our enzymes are going to function and at the rate at which they function. All right, here's a video once again on enzymes just to give you a brief overview. All right, let's look at temperature. How does temperature affect how an enzyme works? So the effective rates on enzyme activity. All enzymes work within an optimum temperature. Optimum temperature means that's the greatest number of collisions between an enzyme and a substrate. For example, our human enzymes work best between 35 to 40 degrees Celsius, which is our body temperature, which is roughly about 37 degrees Celsius or 98.6 degrees Fahrenheit. Now, if I raise the temperature, to boiling point, I can then denature the protein. If I denature the protein, it unfolds and it loses shape. So if I increase my temperature, I cause the enzyme to unravel. If the enzyme starts to unravel, then it can no longer break apart any molecules. However, if I lower the temperature, the molecules move slower. There's fewer collision between the enzyme and the substrate. All right, so let me give you two real-life examples with raising temperature. So if you are an athlete, or even if you're not an athlete, if you're a person that works out or likes to run around, why do you not eat a huge double-double burger before a game? Why would you not eat a huge double-double protein burger or a burger before a game or before you go for a run? So I want you to think about that. I'm hoping you say because if you ate that cheeseburger before you ran on a run or before you played a game, your stomach would become upset and it would probably cause you to throw up. Okay, well, why is that? Well, as you start to run, you start to increase your body temperature. As your body temperature starts to increase at a higher rate, it then disrupts the temperature range to which the enzyme works best. So what happens is I speed up the reaction of the enzyme. As I speed up the reaction of the enzyme, the protein or the enzyme starts to lose shape. If it loses shape, it doesn't function. And in that instance, I wouldn't have the enzymes needed to break up the burger that I just ate. So my body would do what is best and would cause me to throw it up, gets rid of it so that I could wait for my body to go back to homeostasis. Another one is lowering temperature. Okay, an example for this is the reason why we actually put food in the refrigerator. Okay, so if you made dinner or you had leftovers, for the most part, we put all of our leftovers, I'm hoping, inside of a refrigerator. Why is that? Well, lowering the temperature causes the enzymes that break up the molecules inside of the food to work at a very slow rate. So if I put it inside of if I put food within a refrigerator, it will then cause the molecules, the bacteria, and the enzymes inside of the food to work at a lower rate, which would then stop the food from spoiling. All right, these graphs you'll have to know. So let me show you how to read. If I increase the temperature, and then I have the reaction rate. So let's look at human enzymes. As we said, human enzymes work best at 37 degrees Celsius. That's the optimum rate. Notice as I increase the reaction rate or as I increase the temperature, okay, I increase the amount of enzymes that get broken down. However, this here is known as the optimum rate. That is the best rate at which our enzymes work. That's when there's the most number of collisions. Now, as I go past the optimum rate, notice how my reaction rate starts to decrease until it no longer functions, The next one is pH. The pH inside of our system can also affect the rate of reaction of the enzymes. If I change in the pH, I change the shape in the protein. Most human enzymes work between a pH of 6 to 8. It depends on though wherever you are at in your body. So for example, pepsin, which is an enzyme, is found in your stomach. Pepsin works best at a pH of 3. Trypsin, which is in the small intestines, works best at a pH of 8. So let's look at this. So this would be our pH scale. It runs 0 to 14. Notice here at 7, this is optimum for the majority of our enzymes. So we normally have a pH at roughly about 7, okay? So this here is the best rate at which my body works. Now, not all enzymes work best at a pH of 7. Let's look at our stomach. Pepsin works best at around a pH of 2.5. Well, why is that? Well, if you think about it, your stomach is used to basically break down your food. So those enzymes are highly acidic. Because they're highly acidic, that means that the enzyme works best within a pH of less than 7. If we look at our intestines is a little bit more basic. So the enzymes here work best at a pH of 8, okay? Now notice these overlap. Pepsin only works best at 2.5. It doesn't work very well at a pH of 4. And notice that pepsin would be destroyed at anything above 5. Same thing with trypsin. Trypsin will absolutely not work at anything less than 6 or anything greater than 10. All right, here's an example of an egg. And eggs are protein, right? Eggs can denature, meaning that they can change shape. So if I look at an egg, obviously made of protein, right? If I crack it, this is what it looks like on the inside. So if I change the temperature, notice how I change the enzymes, or I change the protein. Now, instead of it becoming liquid, it's obviously more solid. If I change the pH, I can then change the inside of the egg to turn color. If I denature the egg, so I can then add like vinegar. And as we see this occur with our eggs that were placed in vinegar, we started seeing how it started to break apart the shell and the eggs. If I interface, so if I blend it, I change the shape and then pressure as well. All right, this here is our Amoeba Sisters video, which will wrap up the whole unit on enzymes. So I suggest you watch it to help you out. And that is all for today, folks. I hope you learned a little bit about enzymes, about how they function, and what are some items that cause enzymes to denature or change shape.